So hey guys, in this session we'll be going over various important topics related to your theory because your prof exam is coming up, and then I'll tell you important things for the viva, right? The pointers, few pointers, and then in the end I'll be taking a question paper, right? And I'll discuss how to attempt that question paper. So that is a random question paper for final exams I have picked from one of the college, right? So your college might have a different pattern or something, but that way should give you a fair amount of idea, like good idea how to write the exam. Okay? So first of all, we will start with the important topics like chapter wise, but I humbly, this is my request to you, don't just stick. This will give you an overall scenario that these topics are important, but it may change from one college to another because now we are so many colleges are there, different teachers are there, and they emphasize on different topics. So for my students, I always say ki look in the class, what are the topics that were of very a personal interest, right, of your lecturer, meaning in the sense ki unka favorite topic or topic subject kya raha hai, maybe they are doing a research on that. So usually they ask questions from those topics only. So here I'm giving you the general overall view, okay? This is just not a guideline. Don't stick ki isi mein se every question is coming from this only, right? So these are the various chapters that we have done in our class, right? So first is about the subcellular organelles and the cell membrane. So make sure you remember all the metabolic functions of the subcellular organelles, ki what is going on in peroxisomes, endoplasmic reticulum mein, why it is important, what kind of functions or processes are doing, nucleus is important, why, all those things, right? And very, very important are the peroxisomal deficiency diseases, okay? So this topic is integrated to the lipid also, beta oxidation, Zellweger syndrome and all, okay? So you make sure you learn all that. The lysosomes and the related disorders, right? This is also very important because the lysosomal storage disorders, right, we see at many places, not just the eye cell, but the Pompey's disease, which is a glycogen storage disorder, right? And then you have the sphingolipids also, which is also a lysosomal storage. Your sphingolipidosis, those are for lysosomal storage disorders, right? So make sure you go through all of them. Very, very important, right? And for the plasma membrane, you should know the structure of the plasma membrane, its composition, how phospholipids are placed, what kind of movements they are having, various models, what kind of membrane, integral membrane proteins, peripheral membrane proteins, and maybe examples, which we know, adenylate cyclase, 7 transmembrane receptors, right? In the lipoproteins, when you have apoproteins, right, even those are your membrane proteins, okay? Then the various transport mechanism, maybe you can have a short note on the passive diffusion or the active transport, which I'm pretty sure you have done that in your high school also. Okay? So next is about the amino acids. In the amino acid, make sure you know the structures of the amino acid. In few colleges, when I was looking at the previous question papers, they were asking you to know this, and also this comes in the viva. This is a part of a viva also. So make sure you know the structure of the amino acid, various properties associated with it. So best way to learn the properties is in the lecture I have told you this guanidino group is there, right? So those special attachments when amino acids have, they are like 5 or 6 of them. Make sure you pay attention to them, right? So various sort of classification of the amino acid, essential, non-essential, polar, non-polar, all those things, acidic, basic, right? Isoelectric point. I have seen short note of maybe 2 marks or 2.5 marks, right, on the isoelectric point, and the reactions because of amino group. As such, directly we never do this in class, but when we did the urea cycle, I explained you about the transamination, oxidative deamination. Those are the reactions because of the amino group that is present. Also, when you did the degradation, or you can say the catabolism of amino acids. For various amino acid we did one by one. Even that we did, there we did the reactions due to amino group. So transamination, oxidative deamination is very important. Various important amino acid derivatives, right? Like the GABA, ornithine, histamine, creatine is very important. Colored reaction of amino acids are very important, as like it can be a part of your viva also. What are 21st and 22nd amino acid, right? The structure and the function of the peptide bond. You should be able to draw the peptide bond, and what are its properties and characteristics. Ki, is it planar? You can rotate it. What is alpha carbon? Carboxyl किस में होगा? Under various pH conditions, like the ionic form, what it's gonna be. Then you should be knowing about the primary, secondary, tertiary, and the quaternary structures of the proteins, right? So a polypeptide, then the alpha helix, then the beta strands, what kind of helix is there. In general, like small things you should be knowing it, and you should be knowing it like in loop में specifically, either you can have the glycine or the proline, and how this entire the secondary structure is stabilized, whether hydrogen bonding is involved, electrostatic thing is involved. I have given you all the notes. Same is goes for the tertiary structure also, how it is being stabilized. You should know the example of a quaternary structure. We have done the hemoglobin. So you should know the structure of the insulin, hemoglobin, myoglobin for sure. Okay? You should know for isoelectric precipitation is very important. You may have it in theory as a short note, or it can be a part of your viva question as well. So these are the important things about the amino acid. Okay? Now from the enzymes, enzymology, very important. Characteristics of enzyme. Classification is important, right? Coenzymes, cofactor, prosthetic group, what is the difference between them? And remember, you have to know 2-3 examples of each so that you can fill in the page also. Because if it is a short answer question, maybe worth 2.5 marks, in some colleges this question is for worth 5 marks. So based upon that, you just know the difference. First know the definitions, then the difference, and then 2-3 examples, right? The Michaelis-Menten equation. I have never seen, like in the previous papers which I received from students, from you guys, I have never seen any 10-mark question just based on Michaelis-Menten equation. So I seriously doubt they expect you to know all the derivation, but in some college, maybe that is a very favorite topic of your professor. So in those colleges, you know better than me. So make sure you learn it. Otherwise, just know the various steps of Michaelis-Menten equation, how we can do the plot. So if it is worth 5 marks, so you are able to fill 2-3 pages with that. Okay? What are the factors which influence enzyme activities? The temperature, the concentration, concentration of enzyme, concentration of substrate, the pH, all those things. Enzyme inhibition is very, very important. Competitive, non-competitive, reversible enzyme inhibition, right? What are the various plots, right? How Km and Vmax changes upon the competitive inhibitor or maybe non-competitive inhibitor comes in. An example, I have given you the table of the examples also. So make sure you remember 1 or 2 examples for sure. Then plots, definitely you need to know the plot of them, both the Michaelis-Menten version and also the double reciprocal plot. You should be able to figure things out, right? And clinically used enzymes, isozymes, which we have talked about, various form of lactate dehydrogenase, creatine phosphate. That is also, these are important for getting these are early markers, right? For the myocardial infarction, for the pancreatic things, right? So you make sure you do that part as well. Allosteric regulation is very important, right? How it is different than the normal inhibition. What is the difference between allosteric regulation and allosteric inhibition? How the graph is different, because it gives a sigmoidal curve, right? So why it is different, all those things, so that you can write a short note on it. Various marker enzymes are like usually the isoenzymes, but in the cellular organelle, right? The activity enzymes, I have told you that there also. So make sure you know those marker enzymes as well for the various organelles. Okay. Next comes the carbohydrates, a big chapter. Isomerism is important. So know the isomerism, know the various type of isomerism, stereo isomerism, enantiomers, example of enantiomers, okay, so that you can write down a short note on it. The test is very important for the viva or a practical point of view, all the tests that we have done. Mucopolysaccharides, their structure is important, right? Lactose intolerance, glucose transporters, right? GLUT1, GLUT2, GLUT3, GLUT4. So GLUT4 white, GLUT4 is different. What is the special thing about GLUT5? All those things. For the various pathways such as glycolysis, gluconeogenesis, you need to know the organelle, various late limiting enzymes, energetics of it, how it is being regulated. Those things are sure, and I have given you a video also on that, so you can just go and watch that video again, how to learn the pathways. Okay, this can be a short note, hexokinase versus glucokinase, which we have discussed. Fate of the pyruvate. Pyruvate can be lactate, pyruvate can go to acetyl coenzyme, it can be converted back to the gluconeogenesis, right? The regulation is very, very important for all the pathways, right? The Cory's cycle, okay? Cory's cycle and Cahill cycle. Make sure you keep the cycles in front of you and then you compare. Otherwise, many times you have this tendency to get confused between the names because names are very, very similar. So make sure you pay attention to that, right? The Pasteur, Warburg effect, Crabtree effect, right? And I have also discussed you about the Tigar, right? That can be a part of a viva question. Okay, this is usually a viva question, right? The Rappaport Lubring shunt is very, very important, right? Significance of bisphosphoglycerate. This is a part of a hemoglobin also, and this is like an integrated topic between the two chapters of the carbs as well as the hemoglobin. PDH complex, know all the complexes, various enzymes, name of the enzymes, coenzymes, and what are the various other similar complexes like pyruvate dehydrogenase. Okay? The glycolysis, structure of glycogen, 1,6,1,4 bonding, right? Various enzymes which can, like branching enzyme, debranching enzyme, especially how it starts, like when the synthesis starts, right? It starts from the glycogen in that pathway, right? And various glycogen storage disorders also, especially the Pompe and the von Gerke one is most commonly asked. Glucuronic pathway is important. Essential pentose urea. Essential pentose urea is an inborn error of metabolism. So this is also commonly asked. Hereditary fructose intolerance, galactosemia, right? Alcohol metabolism, mucopolysaccharidosis. All these are important topics for the carbs related thing, right? Lipids may know the classification. Blue's classification, okay? And there is always a question, I have seen this is the most commonly asked question about the cephalin, lecithin also. Lecithin is like your surfactant that is related to the respiratory distress syndrome. Sphingolipids, sphingolipidosis, all those diseases you should know, right? And the various oxidations of the lipids, alpha oxidation, beta oxidation, omega oxidation, how they are different, which organelles they are performing, which is the major pathway. Beta oxidation is a major pathway. When omega oxidation will take place, what if the beta oxidation is not working, what is MSUD, right? How the phytanic acid, which is a branched chain fatty acid, how it is going to be degraded. So know all those differences which we have done very clearly. For the synthesis part, fatty acid synthesis part, make sure you look in detail about the fatty acid synthase complex, because I have seen the short note on it. So fatty acid synthase complex is a dimer, how many portion, a condensation unit, a reduction unit, and all that. Okay? and the limiting enzyme, this ACC, right, acetyl coenzyme carboxylase, how it is, and the citrate cycle also, right, because we know that during the fed state we are going to do the fatty acid synthesis, right, and but when the glucose is there, it is going to get converted into acetyl coenzyme into mitochondria. So from mitochondria to the fatty acid synthesis in the cytoplasm, how we transport it, that is also a very important short note. ठीक है, know about the lipoproteins, all kind of lipoproteins, various apoproteins associated with the difference between LDL and HDL is also important, right? Why LDL is a bad cholesterol, why HDL is a good cholesterol. Bile acids and bile salts are important. No primary bile acids, secondary bile acids, right? What if it is not, what if too much of it is gone? What if you don't form the bile acids in properly? Dyslipidemias, right? So hyper or hypo. Hypo may you have the Tangiers, which is very important, right? But know the difference in which you have the highest levels of chylomicrons or in which you have highest level of LDL or LDL receptor related lipidemias is very, very important and commonly asked. Vitamins and minerals, everything you have to run because it is just a standard thing you cannot skip. If you look at the fat soluble vitamins, right, A, D, E, K, vitamin A is important for eyes. There is a question on it. Vitamin D is important. E is also very, very important. K and water soluble vitamins are very important. Even vitamins and minerals actually usually comprise most of your Viva questions, right? The lead poisoning, the arsenic poisoning, right, and the beriberi disease. They can give you the pictures, right, of a patient having the pellagra kind of a condition, so you know it's a niacin deficiency, all those things. So vitamins and minerals, you may see a short note, but most of it would be a part of your Viva questions. Okay? Molecular biology, know the purines, their structures, metabolism, related disorders, right? For the pyrimidine, same, you know the structure, right? Related disorder, Leishman is important, gout is important. Gout is important for the Viva point of view also, right? The orotic acid urea, Watson and Crick model, right? You should be knowing it also. The Chargaff rule, right? Repair mechanism or mutations are important. Topoisomers, topoisomerase, right? That is related to cancer, so that is an integrated topic. Ribozymes is another important. Know the examples, we have done that. Reverse transcription is nowadays important. Reason being, because if you have a question on PCR, if you have a question on COVID, right, so you can say, okay, COVID, then RT-PCR, then you need a reverse transcriptase. So you can use this topic at like both kind of questions, right? Spliceosome, how it is formed, right? Alternate splicing. RNA editing is very important because RNA editing is here also it comes under the lipoprotein part because you have ApoB48, ApoB100, right? So that is an example of RNA editing. So that is very important questions, right? Know the features of the genetic code, Vobel base pairing, degeneracy is important, initiation factors, elongation factors, right? Know the important ones. Which one is using ATP? Which is making a big cluster, which is helping from one place to another place, right? So these small 5-6 points I have given you in the class, so know about them. PTMs, post-translational modifications, gamma carboxylation is very important because gamma carboxylation is a part here also and also you see in the vitamin chapter also. For the blood clotting factors, right? We see do the gamma carboxylation of blood clotting factor 2, 7, 8, 9, right? Chaperones for the protein folding, it is related to the prions disorder, then it is related to the Alzheimer disease also, so you should know the basic as well. Molecular diagnostic and genetic techniques such as blotting, various PCR instruments, that will comprise mostly your Viva questions, right? When the northern blotting is used, for which southern blotting is there, right? So all these things comprise your Viva question, not so asked in theory, but definitely it will help you in the Viva questions, right? Various other miscellaneous topics are the major fuels under conditions in different organisms. So during fed and fasting conditions, right? what your brain is using. Brain is dependent mostly time on the glucose, so is the RBCs, right? So all those things. Glutathione is important. Ubiquitin is important. Urea cycle, they don't ask direct question, but urea cycle you will see in the terms of hyperammonemia. Okay? So hyperammonemia happens when the urea cycle is disturbed. So they may ask you in terms of hyperammonemia or maybe in the glutamate, because glutamate is very important, then maybe asked in the terms of the Cahill cycle. Okay, so know this. And this glycine is a very important amino acid which I have discussed in detail, because it is involved in various pathways for the synthesis of creatine, also for in the heme synthesis, purine synthesis, glutathione, in the conjugation of a bile acids also, right, hyperoxaluria. So pay attention to these features, because they may ask you separately and they may ask you in as a big note on the glycine as well. Serine is important for choline. Choline is an important, right, is a very, very important part. Selenocysteine, right, that is nowadays is becoming very important because of the COVID effect. We know that we need good amount of selenium in the body. Methionine gives rise to cysteine, and then we talked about, right, the folate trap. There can be short note on folate trap, how B6, B12, cystathionine, how it has been controlled, homocysteinuria, right, all those things. Glutamate or glutamic acid is important with respect to hyperammonemia most of the times, okay, with respect to hyperammonemia. So transamination, oxidative deamination explains that. So lysine and arginine is important. These are the basics. So know the structure difference also, various like different R group, what they are called. And another important part is NO synthase that we have done on the topic of the arginine. Branched chain amino acid, 3 of them, but MSUD is the most commonly asked. For the Phe, PKU, tyrosine में tyrosinosis, L-captonuria, albinism, these may be a part of a viva. I have seen that like as a part of viva most of the time. They will show you a picture of a person having the L-captonuria, right? And they will ask you, okay, what kind of disease it is. So you know, okay, the urine is black in color, ochronosis is there, okay, then you will say this is L-captonuria. Same they can do it for the copper. So you will say, okay, this is an Menkes disorder. Okay, things like that. So for the tryptophan, know the pellagra, heart nerve, serotonin, melatonin. Tryptophan gives you so much of those things, right? Histidine, Figlu is important. Figlu is important, right? Because that is related to the folic acid. Citric acid cycle, know the rate limiting enzyme. That is important, right? The anaplerotic part of it, the cataplerotic part of it, right? So when it is doing the synthesis, the first half is anaplerotic. After then it breaks down, things are breaking apart. That is a cataplerotic part. So know all those things. How many NADH, FADH2 are you are getting, where you are getting, what are the irreversible step, that is important. Electron transport chain is important. You should be knowing various name of the complexes, not just the complex 1, 2, 3, 4, but name of the complexes. Again, this can be a part of viva also, right? And you should be knowing the inhibitors of the various complexes, right? You should be knowing about the lead poisoning, cyanide poisoning, right? And then this is ETC is oxidative phosphorylation. So various uncouplers, physiological uncouplers. Okay? And for the heme part, porphyrias are important. That can be a part of viva. They can give you a picture of porphyria and ask you. Bilirubins are important. Bilirubin can be a long answer questions when they can give you certain numbers. They can ask you various kind of questions from this, right? And hemoglobin structure, the cooperativity, how the graph will look like, right? T state, like tout state, relaxed state, right? And various kind of hemoglobinemia. Methemoglobinemia is important, right? Myoglobin structure, they may ask you a comparison between the hemoglobin and the myoglobin. Okay? Liver function tests, all kind of liver functions tests are important. Xenobiotics is important because there is cytochrome P450 involved, right? So read about that, the methanol poisoning, ethylene glycol poisoning, okay, and how it affects the system, right? And in the energy metabolism and nutrition, that may be a part of Viva, BMR, respiratory question, right? SDA, nitrogen balance, protein energy malnutrition, know the difference between the kwashiorkor and the marasmus, right? Why they are different, right? In the kwashiorkor, why you have the edema in one, why you don't have edema in another one, the glycemic index. These are various important topics of under the miscellaneous ones, right? So this is based upon my observation so far, which I have gained from the questions, type of questions you guys have asked me, based upon the previous year question papers and based upon my assessment. So these all topics are to give you a guideline. I'm not giving you a question paper, okay? So this much you should be knowing, and these are very important. Okay? And for the spotters in the lab exam, right? You should be aware of what these instruments are called. I'm not going over all these, but I have given you a slide. You should be knowing a glass rod, how it looks like, the beaker, the conical flask, pestle mortar, the holder, the tongs, what they are called, right? This Bunsen burner is there, and you place the conical flask. Most of the glasswares in your lab is the borosilicate, so because they can absorb the heat very well. They are very good at high temperatures. You should be able to know microscope, identify the microscope also, right? Know these kind of figures, right? The paper chromatography, right? The pH test. I'm not telling you, maybe you can pause the video and maybe come back to the video again and look into the details. So for Viva and the spotters, know this. Urinometer, this is the urinometer, and then you should be able to know about the colorimeter also. Colorimeter has a special small tube that is known as cuvette. That can be of a glass or a plastic, right? So majorly for the students, the glass is given because the plastic one is given because the glasses are very expensive ones. But in the research laboratories, we have the glass cuvettes, okay? And then make sure you know about this albuminometer, may be able to recognize it, right? The vortex mixer up here, you place the small like a cuvette or Eppendorf tube, and then you do the vortex, right? And all the various parts. Semi-automatic analyzer, this is a biochemistry semi-automatic analyzer, right? Everybody knows this water bath, right? You can take this lid out. From here you have the entire bath if you have to put a big piece, otherwise there are small lids, right? This is a water bath system. I'm pretty sure you know about the pH meter, right? This is a typical pH meter, and this is how we keep that in a particular pH solution that is in the beaker. Okay? You should be able to read this graph of OGTT, right? Various serum protein electrophoresis under the normal conditions, maybe they can give you this kind, but at least the normal ones you should be knowing. What is this? This is your electrophoresis thing. See, this is agarose gel electrophoresis. So know all the parts, you should be able to recognize it. And this is what? This is all the tests that we have done in the class, right, for the carbs, right? So they may give you this one, and you have to figure it out, okay, what this test is run for. If there is this violet ring, if you have right, if you have the red color solution, it is red precipitate or a red solution, what does it mean? Know all those things very carefully, right? All the kind of crystals also you should be, these are the Viva questions. Spotters like this one, remember I told you this one is what? Alkaptonuria. They may give you this kind of a thing, this kind of a picture, right? The cardiopedal spasmin tetany, Menkes disorder, right? Kinky hairs. And what is this? This is the normal hand. This is the yellowish thing, that is an iron toxicity. Okay? Arsenic poisoning, this kind of hands, right? Jaundice, you will see the yellow eyes. You should be knowing the normal levels also, right? The rickets, right? They can give you all these kind of images and you have to make things out. The keratomalacia, right? Bitot spots. You see the foamy thing on the white area of the eye. In oil droplet cataract is in the galactosemia. Similarly, I have given you the snowflake cataract also. Kleischer-Fischer ring in the copper accumulation. Gout, why this is happening, you have to figure these things out. And these are few other questions. You should know the cholesterol structure, what is the normal range, know about aldol reductase enzyme, right? Bendix reagent composition. Many a times they ask sulfur containing amino acids are unka test, what are their tests, right? Aromatic amino acids tests, fulfural derivatives are what, right? Liver function test and their normal range. You should be able to calculate the clearance, right, test and calculate the albumin globulin ratios, okay? So these are the various spotters and the viva questions, right? I'm pretty sure you will be able to do it very well. Now, let's discuss previous year annual exam discussion. Every college has a different pattern. I have randomly picked one, right? And in earlier also I have released a video in which I have told you in general how to attempt the exam. Multiple choice question, do first because every question is giving you 1 mark. And that will take small amount of time. So do that first. Then you shift to the short answer questions, 2 mark questions, right? Because if you do it right, there are higher chance examiner is going to give you full point. The bigger questions like 10 mark questions, in some of the colleges they have the 5 mark as well as the 10 mark questions. Do them in the end because 10 mark question is going to take lots of time, right? So let's say you have done with the entire paper first in let's say 2 hours, then you have entire 1 hour just dedicatedly for that question, to attempt that question, maybe 1 or 2. So you are not worried about कि मेरा question paper will be left, ठीक है, वो छूट जाएगा, ऐसा कुछ नहीं होने वाला है. So multiple choice question, short answer questions, then shift to the long answer questions, ठीक है. So this is a question paper, right? This is the annual one for 3 hours, right? And all questions are compulsory in this. All questions are compulsory. Okay. So section 1, section A is what? Section A is MCQs, which I don't have. So whatever it was, I'm pretty sure you were able to do it because that was separate sheet given to the students. So section B comprised of 40 marks had a question. What was this question? Is this question we have a clinical case scenario. A 35-year-old woman presented with a history of a fever for a week, malaise, nausea, loss of appetite, pain in right hypochondrium. On examination, she was found to be icteric. Okay? Icteric means what we are looking at? We are looking at the jaundice. We are looking at jaundice. ठीक है, ये final है. And the liver was enlarged and tender. Following are the results of various lab tests. Serum total bilirubin was 5.2. Indirect bilirubin was 3.7 and direct bilirubin was 1.5. Remember I told you you should be knowing the normal values. So these are the normal values. If you look at the normal values, right, the total bilirubin level is high. Both indirect and direct bilirubin levels are high. So what is the probable diagnosis of this case? Whenever you have such kind of a condition, we know there is a problem with the liver. Right, there is a problem with the liver functioning, right? So it is a jaundice due to hepatic parenchymal disease, right? So see, bifurcation is 1, 2, 2 and 5. So if you will just write this is an hepatic parenchymal disease, it should be enough. But you can also write since the serum total bilirubin is raised because both the direct and indirect bilirubins levels are increased, right? So hence you concluded that there is a hepatic parenchymal disease and probably the stools and the urine of the patient is normal. The color has not been changed and you should get a full 1 mark for that. So now next question for this is which is worth 2 marks, right? 2 to 5, 2 marks. What are the other tests you will do to confirm the diagnosis, right? We know that the gold standard is the liver biopsy. Gold standard is the liver biopsy. You can write that down, but we can do enzymatic ALT assay also. We can do enzymatic ALT assay also. Good. So that will give you full marks, right? What can be the possible cause leading to this condition? What can be the possible cause leading to this condition? ठीक है, क्यों ये हुआ होगा, right? Mainly because of viral hepatitis. Why I am saying viral hepatitis? If you go back to the question with the history of fever for a week, ठीक है, right? So probably the patient was having viral hepatitis. That can be hep A, B, C, D, E. You can write all of them. The reason may be of the closed contact, maybe of a blood transfusion for the sexual contact, right? Because there was hep A, that's why hep D also came in. All those things you can fill in and you can get 2 marks. Distinguish between various type of jaundices, right? Obstructive jaundice, neonatal jaundice, right? If you can recall, in the class we have done this table also, right? Hemolytic jaundice, obstructive, hepatocellular, neonatal jaundice, right? So you can write maybe 1 or 2 pages to get full 5 marks, or maybe draw this table, right, to attempt this part, distinguish between various type of jaundice based upon which bilirubin, direct or indirect bilirubin, is high or low. Okay? So that will give you full 5 marks for this question. Now the next question is explain why. There are 4 questions, each worth 2.5 marks, right? So it means you have to make, maybe depending upon how big your answer sheet is, maybe fill 1 or 1.5 or 2 pages, like front and back. As sunlight is essential for bone health, right? When we are talking about bone health, we are talking about the calcium. Why we need sunlight for the calcium? Remember when we did this vitamin D thing, this chart? Skin you have, in the skin what you have a special kind of a cholesterol, that is 7-dehydrocholesterol, right? This is from the class. So you can explain this chart. That will give you full marks, because ultimately this calcitriol will be helping in maintaining the calcium levels, and that will help in maintaining the bone health. Okay. Now, short chain fatty acids are prescribed in patients having carnitine deficiency. What is carnitine deficiency? Why it is important? If you remember from the lipid chapter, right? When we have to do what? When you have to do the beta oxidation, right? So you have acetyl-CoA. You have acetyl-CoA here, which should pass through the outer mitochondrial membrane and inner mitochondrial membrane to reach to the matrix, right? And there is the place where your beta oxidation is going to happen. That's how you, sorry, not the acetyl-CoA, but the fatty acid. Fatty acid, and here via the beta oxidation you will get the acetyl-CoA. Okay? So fatty acid cannot pass through except the short chain fatty acids and medium chain fatty acids. Less than 8 to 12, between 8 to 12 carbon long fatty acids can easily pass through and carry out the beta oxidation. But for fatty acids longer than 12 carbon long, they have to be carried, we have to make them pass through the membrane. For that we know we have the CPT1, CPT2. And there is this carnitine that is involved, if you go back and look. Right? So in case of carnitine deficiency, you cannot transport these long chain fatty acids into the mitochondria for the beta oxidation. Right? So how you are going to get the energy? That is why the patients are supplemented with the short chain and medium chain fatty acids. Right? So you have to explain all that here in the carnitine deficiency. Okay? Now, glutamate plays a role in amino acid metabolism. What we are talking about here? We are talking about here transamination. transamination and oxidative deamination that we have done. Usme what we have done, we know there is glutamate. This glutamate will produce alpha keto acid. That involves a ammonium ion, which can be taken or which can be released to the system. So that entire part where we have done about ALT, AST thing, you have to produce it here. 2.5, you will get the full marks. Question: edema is seen in kwashiorkor, not marasmus. Why is that? Because we know that kwashiorkor person is deficient in the protein, whereas marasmus person is deficient in both the carb and the protein. Okay? When you have the less amount of protein, then the amino acid pool is disturbed. Then the albumin formation is disturbed. If you don't have enough of albumin, that will lead to the edema, right? But whereas in the marasmus, both pools are disturbed. You have distorted liver amino acid pool and also the caloric pool, right? So everything is disturbed, which will lessen the lipoprotein synthesis, and that will give you the fatty liver, not towards the edema. Okay? So that chart you have to reproduce it here. 2.5 you will get. Now there is third question, 5 plus 5. So you have to write maybe good 2-3 pages to this. Role of cholesterol in the body. How you are going to fill 2-3 pages. What do we know about the cholesterol? We know the structure of cholesterol. We can write it down, right? So for this, let me take you to the book. This is which book? This is the Vasudevan book. Okay? In this Vasudevan book, why I am telling you this? Because I want also you to be very independent, so that you can browse through the book and you should be able to figure out ki answer kaise find karna hai, how to find the answers from the book. So this is the cholesterol chapter. We have to write down the importance of cholesterol in the body, where it is being used, for 5 marks question, 5 points lene hai. And this is the entire chapter. We cannot reproduce the entire chapter. We have to just fill in 2 pages or 3 pages, enough, this much enough कि 5 points तुमको मिल जाएं. So what if I am you? तो मैं क्या करूँगी? If I am you, then what I will start is, I will start with the cholesterol का structure. Okay. So what I will start is why this is called cholesterol, what kind of cholesterols are there, okay, from the introduction. From the introduction, I will do that. Okay. Cholesterol is a light yellow crystalline solid. The clinical significance of cholesterol in the heart disease, maybe 1 line for that. Then I will try to reproduce the functions of the cholesterol. Do you see here the functions of the cholesterol is given here? This function of the cholesterol is given here. Okay? Maybe write that and then maybe produce a structure of the cholesterol. Structure of cholesterol. Okay? And then maybe in bullet points I can tell how this cholesterol is being synthesized and what is the rate limiting enzyme, and then cholesterol is not water soluble, and if you have to remove the cholesterol from the body, you convert it into bile acid. Just 1, 2 line of all these things should be more than enough to give you the 5 points in the exam. Are you getting me? Ammonia toxicity. How will I attempt this ammonia toxicity? That is worth 5 points. I will start with this fact that ammonia in your body is coming from the amino acids se aa raha hai. From the dietary protein, you are breaking these things down. So I will start from the transamination, oxidative deaminations, which we have done in the urea cycle. Then I will explain the role of glutamate there. Okay? And then maybe a urea cycle ka pura cycle bana ke, then I'm going to tell that this thing entire cycle is taking care of the ammonium ion. If there is any disturbance, that will lead to elevated levels of ammonium ions, because all the urea cycle disorders have hyperammonemia, right? If it gets accumulated in the body, that is going to increase the blood urea nitrogen. Okay? All these things should suffice the ammonia toxicity. And then you may be able to explain if there is too much ammonia in the brain, right, that is going to lead to the edema in the brain also. 1-2 lines of that you can add. 1-2 lines of that fact you can add, that since we know that glutamate is the one, alpha ketoglutarate takes up one ammonium ion and becomes glutamate, and it can take up one more ammonium ion to become glutamine, right? So in this ammonium toxicity or in the hyperammoniamic condition, what happens is there is decreased level of alpha ketoglutarate, and this alpha ketoglutarate will not be available for the TCA cycle. That will make the person exhausted because there is no ATP that is being released. So all these information should suffice the interest of the examiner, and you should be able to get good marks. Okay? Now there is another question of the short note, and that is like there are 4 questions worth 2.5, 1.5, 2 pages should be enough, I guess. Glycemic index, I have told you in the theory part also, you should be able to write down the note on the glycemic index. How you are going to write it down? Again, going back to your book, that is the Vasudevan, which most of you have it, okay? And the page number is chapter 40th it is. Chapter 40 mein hum jayenge. Here we go, and then you can figure out this is the chapter on energy, metabolism and nutrition. Okay? So there you can first of all tell an introduction about this part here. You may not be able to read it properly, but you may be able to make it out. This is 528 page of Vasudevan. There they are talking about the energy requirement of a normal person. कि एक normal person का energy requirement is what? ठीक है? And then you can talk about कि if person has a sedentary lifestyle or if it is a ruksha puller, what kind of lifestyle changes the need. Then you can talk about here the importance of carbohydrates. Right? Because we are coming to the glycemic index. Glycemic index is related to the carbohydrates. How much carbohydrates are needed, and 1 gram of carbohydrate gives you how much of the energy that you can tell, right? So there, and then and then on page number 531, when you will talk there, there is a small topic on the dietary fiber. Okay? There is a topic of the dietary fiber. In that dietary fiber topic, right, you have this hypoglycemic ka, and then there afterwards what you have is you have a table which tells you the glycemic index of various foods that you eat, the cane sugar, rice, wheat, bread, and that should suffice the like 5 marks which you want. Itna likhoge, that should be more than enough for the 5 marks, okay? Coming back to the next question, that is competitive inhibition of the enzymes. Very easy. We have done this in the class also, if you can recall. This inhibition, enzyme inhibition, you can write down what an enzyme inhibition is, what is an enzyme. There are various kind of enzyme inhibitions. You can make this and also write a paragraph to fill in the pages. You have reversible, irreversible, allosteric inhibition, competitive and non-competitive are the reversible inhibitions, right? Then you can give this graph also in 2-3 lines, explain the graph also. Then you can give the examples, few examples, not all of them, but the few examples also. Then you may be, if you want, if that pages are not filled, you want to give little bit more to get full marks, then you can give also the Lineweaver-Burke plot. That should satisfy the examiner in relation to this competitive inhibition of enzyme. Respiratory distress syndrome in newborn, you have to tell about the lecithin, dipalmitol, why it is important. We have discussed that in the lipids chapter. 2.5 jitna humne kiya hai, that should be enough. Cytochrome P450, xenobiotics, we have done that, right? And also if you go back to the book again, right? If you go back to the book again, the detoxification and the biotransformation of the xenobiotics, here when you scroll down on the second page, there is this cytochrome P450 enzyme systems, right? This is Vasudevan book. So you can just pick out the major points from here and also maybe from the class which I have given to you. Very easy to attempt. Now next question is question number 5. A 50-year-old man presented with the complaint of excessive urination, increased appetite and thirst, and diagnosis was made diabetes mellitus. What is the diagnostic criteria of diabetes mellitus? We know we are going to look for the glycated hemoglobin because too much glucose is available there. OGTT test, goat pot test, that is an enzymatic method. You can explain that because this is worth 4 points. That should suffice the cause. और उसके बाद question is explain the biochemical basis of the signs and symptoms of the diabetes mellitus. We know what is 3 P's are there: polydipsia, polyuria, polyphagia. Excessive hunger, excessive thirst, and excessive urination, right? And why that is happening? Because we know, we have done this in class also, that if you have glucose levels elevated for the prolonged period of time, that triggers your MEOX, that triggers your myo-inositol oxygenase activity, which increases the myo-inositol catabolism, which alters your sodium-potassium mechanism, ATPase mechanism, and that you have done in the physiology also. If that is disturbed, right, which includes the secondary signaling pathway, it results in diabetic neuropathy, diabetic retinopathy, diabetic nephropathy, and the diabetic cataract also, right? And in the uncontrolled diabetes, because of elevated level of glucose for the prolonged period of time, that increases the osmotic pressure also, which leads to the impaired vision also because it affects the retina as well. And also because of the elevated level of glucose levels for the prolonged period of time, you can say that because of the Maillard reaction, there will be increased levels of the superoxide, right? And that is going to affect the intelligence, the memory, all those brain system will be affected. So that should give you the 3 points for this question. This is 3 marks again. Enumerate the long-term complication that can occur to diabetes mellitus patient and discuss what will happen. Dyslipidemia. Q, glucose level is low. Hypoglycemic condition is there inside the cell. That will lead to increased beta oxidation. ठीक है? Okay, ये थोड़ा delete करेंगे. So for the long-term complications, right, what we can introduce is the dyslipidemia. Why? There is no glucose available in the cell. There is glucose, but not for the cell. So that will lead to what? That will lead to the movement of the increased, you can say, the free fatty acid oxidations. ठीक है, फ्री फैटी एसिड्स बहुत सारे हो जाएंगे बिकॉज नाउ द ट्राइग्लिसराइड लेवल्स pools are being disturbed unable to produce the energy. So there will be like so many free fatty acids. Few of the free fatty acids will help you to like nurture your body, nurture your cells, but there are excessive free fatty acids available which cannot be controlled by body, which will lead to what? Increased triglyceride productions, right? And that increased triglyceride in the system, in the plasma, right, free floating plasma will lead to increased ApoB, will lead to increased VLDL, right, increased HDL levels, right, and that will lead to the cardiovascular diseases also. If you will write this much, you can get 3 points. Explain why night blindness occurs in vitamin A deficiencies. You have to just visual walled cycle you have to explain, and that should give you 2.5 marks. Cyanide poisoning, we know the cyanide is gonna go and bind to the cytochrome B, right, which is a part of your electron transport chain. It will come to halt. There will be no oxygen available to the hemoglobin also, because it binds to the hemoglobin also, so that it enables the hemoglobin to take up the oxygen or release the oxygen. It's just stuck there. But the major thing is the electron transport chain, the complex 4. ठीक है. Chronic alcoholism lead to fatty liver, right? Because when you know about the alcohol is converted into aldehyde, and that process we are releasing lots of NADH. ठीक है. If NADH levels are very, very high, that is going to decrease the gluconeogenesis. That is going to decrease the beta oxidation. Right? Gluconeogenesis कम होएगा तो what will happen? It will lead to the increased level of the pyruvate. Pyruvate will be converted more into acetyl-CoA. ठीक है? A beta oxidation, gluconeogenesis is halted, acetyl-CoA is elevated, so acetyl-CoA will do the fatty acid synthesis. That is why it will lead to the fatty liver. ठीक है? HDL is a good cholesterol. Why HDL is a good cholesterol? You can explain the structure of a HDL, what kind of apoproteins are in there, and then you can explain how via the ABCDA1 transporter protein, it takes up the cholesterol from the peripheral tissues and it delivers it to the liver. If you will explain that mechanism, that should give you 2.5 marks, more than enough. Differentiate between enzymatic, non-enzymatic antioxidant. Enzymatic antioxidants are your glutathione, superoxide dismutase, all those. They need the coenzymes. There are specific places that is happening. They work with the hydrogen peroxides and reactive oxygen species. Whereas non-enzymatic antioxidants, they do not need a complex mechanisms. They do not need the enzymes, coenzymes, right? And there are of two types. We know that there's a natural and the synthetic. Natural ones are your vitamins, vitamin C, vitamin E, right? Vitamin K. These are your natural antioxidants. And if you will write two lines from where they are coming, which sources are there, you should be good enough. Enzymes are proteins. These vitamins are your non-enzymatic, and that vitamins you consume in your diet. Okay, vitamin C is water soluble, but the other two are the non-soluble ones. Okay, enzymes, you don't take it in diet, it is present in your body, it is synthesized by your body itself. So these kind of differences should solve the problem. Liver and the glycogen, muscle glycogen metabolism. Okay, so we know the glycogen metabolism from the class also, right? You can explain them. The difference between is the enzyme phosphorylase. When you break down the glycogen, you have the muscle phosphorylase, you have the glycogen phosphorylase. Those two are different. Then the diseases, the McArdle disease is because of the problem with the muscle phosphorylase, whereas hers is the liver phosphorylase deficiency. You can tell about that. Then you can compare it with the Tourette disease also, and then you can tell that in the muscle muscle stores less of the glycogen, most of it is in the liver. Muscle glycogen is usually stores are used during the excessive exercise. All those sort of things you can tell. write short or not on factors affecting iron absorptions, right? So iron absorption is increased or decreased by various factors, right? How it is being increased? The vitamin C, your meat, your poultry, they increases the iron absorptions, whereas the tea, coffee, right, all they decrease the, means they are, which one are having phytanic acid, they decreases the iron absorption, right? Regulation of phosphofructokinase, very important. I keep telling you all the time, and also in the revision also, in the doubt solving session also. This is from the Harper book. This is from the Harper book, right? This is figure 19.4, chapter 19. You should be knowing this one. How in the gluconeogenesis and the glycolysis you are regulating this phosphofructokinase, right? Gluconeogenesis is not the exact reversible of glycolysis, but their few parts are common. This is a biphasic enzyme, right? It has two phases, right? One phase works during the gluconeogenesis condition, one phase works during the glycolysis condition. And this is the entire regulation. If you make this kind of a figure and just write few lines afterwards, that should solve the problem and you should be able to get the full marks, because it is just 2.5 points ka hai. Okay. Rickets, we know, calcium deficiency, right, that gives you rickets. In the kids, that gives you the rickets, right? And also, you know a lot, I'm pretty sure this is very easy for you to attempt. Role of HMP shunt in your body. You can give all the names of HMP shunt, which organ and organelle it is performing, right? And then you can say it is an oxidative phase, non-oxidative phase. Oxidative phase gives you what? Non-oxidative gives you what, right? And that should be more than enough for the HMP shunt. When HMP shunt is taking place, you can also tell that when you have too much of the glucose, right, and glucose cannot go via the glycolysis, it will be shunted towards the, it will be shunted to the HMP shunt. During the cell cycle, cell division also, HMP shunt will be happening. It gives you NADPH also, right, which is needed at various places. That if you write all those lines, that should give you 2.5 marks. Right, and that is pretty much about this question paper, and I'm pretty sure now you will be able to think how you have to attempt the question. You know a lot, all you have to do is bring in the pieces from the various chapters from the several topics and then make a good answer, frame a good answer out of it. Framing the answer is important and that will help you to get the good marks. Highlight the points, I always say, make the bullet points, make the graph, make the chart, and then explain that in a paragraph form also. That will fill in the page also, and the teacher will get a good idea that you know the concept, right? With this, I will take a leave and thank you very much. If you have any questions, you have my email ID, you have my Telegram group, and you can contact me on the app as well. So good luck to you and be happy. Just be happy, because if you are happy, your brain is going to work more. So study smart is important, right? Just don't work overnight. Little pieces every day you do, small topics, and you are going to do great. Thank you.